Okay, let's go ahead and get started. And this is chapter 11. Okay, it talks about this first part just talks about some of the general overview. And so we're scoping out the chapter, the basic goals of antimicrobial control. So you have antimicrobial means against microbes. Anti is against. Microbe refers to living. So it's against microbes and includes any method for keeping undesirable, harmful microbes out of the wrong places. Control agents are applied for sterilization, disinfection, and asepsis. We'll go through all of those slide by slide. So we're going to talk about physical methods of controlling microbes, and that's heat that is wet or dry. So applying increased temperature or heat is the most common method of achieving microbial death. Heat can be applied in combination with water, which is called moist heat, or water-free, dry heat. Then we have agents of microbial death. Well, how do they work? So you can have physical, which is energy-based, and chemical, which is molecular-based. We'll go through those. Those agents are directed against microbial structures and functions and designed to block their reproduction and survival. And then we're going to talk about physical methods. Zapping with radiation. Its high energy wavelengths such as gamma rays, X-rays, and even ultraviolet rays can sterilize and disinfect materials without using heat. Then we have chemical methods. So you have molecules that destroy microbes. So a wide range of chemicals can kill or inhibit microbes on inanimate objects or the human body. Inanimate means not living objects or on the human bodies. So we can have disinfectants, antiseptics, sanitizers, or degermers, and we'll talk about each one of those. And to finalize this chapter, we're going to talk about common chemical categories. So antimicrobial chemicals come in the form of liquids, gases, or solids. Classes of chemical agents include chlorine, iodine, alcohol, phenols, peroxides, aldehydes, gases, detergents, and soaps. And we'll talk about each one of those, and I will tell you how much in each of those that I need for you to know for the quiz. Okay, controlling of microorganisms. So controlling exposure to potentially harmful microbes is essential to our lives and has been a concern also in the past. So early protection attempts laid the foundation for microbial study methods today. So in the past, before they even understood how microbes work, they would actually burn wood. It releases formaldehyde, and that would actually kill some microbes. Different herbs, perfumes, vinegar contain mild antimicrobial substances. Here's an interesting picture. You'll see this in the book, and it shows this is a doctor, and a doctor that was garbed up, and he was treating a patient that had a plague. And so you see he was protecting every aspect of his body. And the reason he had his face covered with a beak is in that beak, there are a lot of fragrances that they noticed that would deter microbial growth. So general considerations in microbial control. First, you have physical, chemical, and mechanical methods to destroy or reduce undesirable microbes in a given area, which is decontamination. So the primary targets are microorganisms capable of causing infection or spoilage. So first you have vegetative bacterial cells. Vegetative means they're actively dividing, they have metabolism, they're living. And then also you have endospores. That's a very resistant form of the microbial, that when there's not enough, when it's not conditions that are good for their growth, they can go and create endospores, which they could survive for a long time in extreme conditions. Then you have fungus. So you have fungi, hyphae, and spores. You have yeast, protozoan, worms, viruses, prions. Those are the different types of microbes that we target. So some of the methods that we have are physical agents. So with heat, we have dry heat, like incineration. With incineration, we can actually achieve sterilization. You have dry ovens, which you can achieve sterilization. And once again, we'll go over these more in depth as we go further along. Then you have moist physical heat, and that's a physical agent, and that's steam under pressure. You can achieve sterilization. You have boiling water, hot air, pasteurization. You actually achieve disinfection and not necessarily sterilization. We'll go through the definitions of those in the next few slides as well. Then you have radiation, like ionizing radiation, like X rays, gamma rays. You can achieve sterilization, and ionizing radiation. It's like UV light, and you can achieve disinfection. Some of the mechanical removal methods, filtration, like you have air filtration, that you can get some disinfection. Then you can have liquids that would be, uh, you can achieve sterilization. And chemical agents or some gases can have sterilization and disinfection. Then you can have liquid microbial methods, and you can have liquid on animate, which is living substances. So you have antisepsis, and then you have inanimate, and that's on non-living surfaces. You can have disinfection and sterilization. So here's the definition of disinfection. Disinfection is the destruction or removal of vegetative pathogens, but not bacterial endospores, usually used only on inanimate objects. So disinfection or removal of vegetative. So those are the living, the ones that actively divide, binary fission, the ones that actually have metabolism, everything that you would think of in terms of them being alive. That's disinfection, but it's not usually disinfection. This means it does not kill the endospores, bacterial endospores. Then you have sterilization. That's the complete removal or destruction of all viable microorganisms. So this is typically used on inanimate objects. So with sterilization, kills the bacterial spores, kills the endospores, kills the vegetative pathogens, kills everything. That's sterilization, as opposed to disinfection, typically does not remove bacterial endospores. Antisepsis, that's where you have chemicals applied to the body surfaces to destroy or inhibit vegetative pathogens. It's antisepsis. So here's some of the relative resistance of microbes. In the highest resistance, you have prions, which prions are very interesting. They are very hard to kill. They are basically just portions of protein that can infect a cell. They're not considered living, but they can infect the cell, and we'll talk about them a little bit later, and they're very hard to inactivate. Then you have bacterial endospores that are very resistant, hard to kill. Then you have moderate resistance. You have naked viruses, protozoans, bacteria with no endospores, but resistant walls. They have moderate resistance. Then you have the Mycobacterium, Pseudomonas, Staphylococcus, and then most heat and chemical resistant bacteria have moderate resistance. Then the ones that have the least resistance are the vast majority of bacterial vegetative cells, bacteria that is actively dividing and has active metabolism. They're the least resistant. Fungal spores and hyphae, yeast, the envelope viruses, they're least resistance as well, and the protozoan and trophocytes. So here's some of the relative resistance of bacterial endospores and vegetative cells to control agents. So in this, it kind of says, if something has the endospore, as an endospore, this is about what level you have to heat it up in order to kill it. That same organism, if it's in vegetative form, this is about the temperature you'd have to heat it up to kill it. Relative resistance, like least resistant, most resistant, extremely resistant, this is the 1.5 times. So most organisms with endospores, vegetative form, to heat are considered 1.5 times resistant. Radiation for endospore takes 4,000 units as opposed to 1,000 units for vegetative, sterilizing gas or sporicidal liquid. So it shows the differences if it has, uh, if they have endospores, and then that same organism if it was vegetative. So like in this, if you had a something that could kill spores, a liquid that could kill spores, you'd have to have it soak for 3 hours in order to kill the endospores. But you only would have to let it soak for 10 minutes if it was just vegetative forms. Okay. Then we're going to talk about, that's where we're talking about resistance of endospores. This is an organism that has an endospore. Okay, this terminology and methods of control. So, wait a minute. Okay, so now we'll talk about terminology and methods of control. So sterilization, process to destroy all viable microbes. Then the microbial agents are the germicides. So you have antimicrobial agent aimed at destroying a certain group of microorganisms like bactericide, fungicide, virucide, sporocidal. So if you see side, it's talking about killing it. So with disinfection, you have use of physical process or a chemical agent, disinfectant, to destroy vegetative pathogens, but not the bacterial endospores. That's disinfection. Antisepsis is application of chemical agents directly to exposed body surfaces, wounds, and surgical incisions to destroy or inhibit vegetative pathogens once again. Notice the difference between sterilization. It destroys all viable microbes, including the spores. Disinfection kills the vegetative pathogens, but not the bacterial endospores. Antisepsis kills the um, bacterial, vegetative bacterial pathogens, but it does not kill the endospores. And it's directly on exposed body surfaces. So the agents that cause microbistasis, that's antimicrobial agent aimed at temporarily preventing microbes from multiplying. So this is as opposed to microbistatic, um, which actually it doesn't kill it, it actually prevents it from multiplying. So actually, let's go with terminology and methods of control. So you have sterilization, which is process to destroy all viable microbes. Then you have microbial agents, which are antimicrobial agent aimed at destroying a certain group of microorganisms. And so then you have of microbial agents, you have disinfection, which is use of physical process or chemical agent or disinfectant to destroy vegetative pathogens, but not destroying the bacterial endospores. Antisepsis is application of chemical agents directly to exposed body surfaces, wounds, and surgical incisions to destroy or inhibit vegetative pathogens. So know the difference between sterilization, that's destroying all viable microbes, including the endospores. Disinfection is destroying vegetative pathogens, but not the bacterial endospores. Antisepsis is chemical agents that directly put on exposed body surfaces, and it inhibits or destroys vegetative pathogens, but not uh, the bacterial endospores. Then you have agents that cause microbistasis, which is antimicrobial agent aimed at temporarily preventing microbes from multiplying. So the difference between microbistasis and microbicidal. Microbicidal is it kills. Microbistasis, it slows it down, temporarily prevents them from multiplying. So there's some methods that reduce the numbers of microorganisms. So the total numbers, you have sanitation, it's any cleansing technique that removes microorganisms from inanimate objects. Inanimate means non-living surfaces to reduce the potential for infection and spoilage. That's sanitation. That will help with microbistasis. It's not a microbicidal. Then you have degermination, and that's reduction of microbial load from living tissue by mechanical means. So, of course, that's a fancy name for washing your hands. But any living tissue that you reduce the microbial load is degermination. Here's some of the table that shows microbial control in terminology. And you have decontamination, destruction, removal, or reduction number of undesirable microbes. Some examples: disinfection, sanitation, degermination, sterilization. And you have sepsis, which is the growth of microorganisms in tissues. Asepsis, techniques that prevent the entry of microorganisms into sterile tissues. Antiseptic, chemicals applied to body surfaces to destroy or inhibit vegetative pathogens. Then you have disinfection, destruction of vegetative pathogens on inanimate objects. Sanitation, cleansing techniques that removes microorganisms and debris from inanimate objects. Degermination, cleansing technique that removes microorganisms and debris from living tissue. Then you have sterilization, removal or destruction of all viable microbes. Important to know these terms, both for this class and in general. You need to know these terms and what they actually do. And here's some examples of each of those. Like you see, sanitation's dishwashing, laundering clothes. Degermination is surgical hand scrub, alcohol wipes, hand sanitizers. Okay. Microbial death. Microbial death is hard to detect because microbes often reveal no conspicuous vital signs to begin with. So a lot of microorganisms, they don't really look like they're alive. They don't seem to have a circulatory system or anything that we can monitor that say they're alive. So permanent loss of reproductive capability, even under optimum growth conditions, that's how we will define microbial death. So if it has permanent loss of reproductive capability, even under optimum growth conditions. So the factors that affect microbial death: the number of microbes that you're starting with will affect microbial death. The nature of microbes in the population, which type of microbes you have, that will affect death. Temperature and pH of the environment will affect microbial death. Concentration or dosage of agent. So what the agent is that we're using to cause microbial death, the concentration or the dosing of it will affect microbial death. Mode of action of the agent, how the agent actually goes about killing the microbe. Presence of solvents, organic matter, or inhibitors. If there's anything else in the solution that could hinder the agent's working. Okay? And that, organic matter may be something as simple as you have saliva in your mouth, and so the microbial agent you put in there to kill the microbes on your teeth could be diluted out or counter-affected by the saliva. It's organic matter. So here it talks about the factors that affect death rate. So here, this is the log number of viable cells. The higher it goes, the more viable cells you have in solution. And this is over time. So you put a certain amount of antimicrobial in with the viable microorganisms, and it starts off very high. It's a whole lot. And the longer it's in with the antimicrobial, you have fewer and fewer of the viable microbial cells. And then it gets to a certain point where it's clear, and it's cleared of all the microorganisms, that's the point we call sterilization. And so this is it just talks about the load. Once again, it's like if you have a low load, low number of microorganisms, and a high load, how long it would take before you achieve sterilization. If it's a low load, it takes shorter time to get to no microorganisms. A high load takes a longer time. Then if you have spores present, or you just have vegetative cells, if you have just vegetative cells, no spores, it doesn't take as much time to kill them all off if they're uh, if they're associated with an antimicrobial agent. It doesn't take as much time. If spores are present, or spores are present along with the vegetative cells, even after the vegetative cells are killed, and they're still exposed to this antimicrobial agent, over time, you have fewer and fewer spores, but it's very, very hard to get rid of the spores completely where you have complete sterilization. And this is if the number of viable cells, microorganisms, so it's high, and it just keeps staying high, you add an antimicrobial agent, and then it doesn't take much time at all, where the number of microorganisms, it goes down tremendously. But if you take this antimicrobial agent away, then the viable cells that are left will go back up to the original number. That's where it talks about microstatic agent. That has something that increases the microbials to actually grow back to where they were. That's if the microbicidal is taken away. So practical concerns in microbial control. First off, the selection of method of control depends on certain circumstances. So does the application require sterilization? Like, does it need sterilization or just disinfection? So the item that you're trying to sterilize, is it going to be reused or are you going to throw it away? Can the item withstand heat, pressure, radiation, or chemicals? And is the method used to sterilize it suitable? Will the agent penetrate to the necessary extent? And is the method cost and labor efficient? Is it safe? So when they're talking about microbial control, they're saying like you have surgical equipment. So you have scalpels and you have catheters and things when you're going into aseptic area inside a person's body. You have to make sure that there are no microorganisms that you're introducing into the body cavity. So the tools, the things that you use in the room, they all need to be sterile. So modes of action. So these are the antimicrobial agents' modes of action. So some of them have cellular targets of physical and chemical agents. So some target the cell wall. Cell wall becomes fragile and the cell lyses or bursts. Some of them act on the cell membrane. It means that they'll lose their integrity. The microorganism cell membrane loses integrity. If it loses integrity, then it will not be able to maintain its internal environment. And so here's where it's like, this is a antimicrobial, and it goes down into the cell membrane, and it causes the cell membrane to start breaking apart. Once it starts breaking apart, then the cytoplasm will start leaking out. Other things from outside will leak into the microorganism. Then you have cellular targets of physical and chemical agents. So if it doesn't attack the cell wall, cell membrane, then you can have it where the antimicrobial attacks the protein and nucleic acid synthesis. So it can prevent the replication, transcription, translation, maybe peptide bond formation, all the things that are utilized in protein synthesis. The antimicrobial could actually keep the microorganism from being able to repair itself. That's how it acts. And then you can have modification in how the protein functions for the microorganism. So it disrupts or denatures proteins. Okay. So here's if you have an antimicrobial and it's acting on a protein in a microorganism. So this is let's say this is an enzyme. It's a protein in the microorganism. It has a substrate what it acts on, and this is uh, the tertiary configuration of an enzyme. So if you add heat or you change the pH dramatically, you may be able to denature this completely. And so instead of having this tertiary structure, it goes back to primary or secondary structure. But either way, it won't be able to do what it was made to do. Then if you have some heavy metals, that the heavy metals uh, block the active sites where the substrate could actually act. And so if it blocks the active site where this substrate would have come in, then this enzyme will not be able to do what it's supposed to do in a bacteria or in a microorganism. Then you can actually have with heat or pH change, if it didn't completely denature it, it can also just change its shape. And if it changes the shape, so this is the shape that's needed for act on this substrate and this substrate. If you change the shape enough where now it doesn't act on these substrates that it was made for, once again, it deactivates it. So all of it comes back to this. The active site can no longer accept the substrate and the enzyme is inactive. Okay, if you wipe your hands with the antimicrobial cleanser, you're accomplishing what? Right? Antisepsis. Very good. Okay, here's some of the physical methods of control. So the vast majority of microbes are readily controlled by abrupt changes in their environment. So some physical methods are heat. If you abruptly change the heat, and you can have moist or dry heat. Sometimes cold temperatures can kill microbes. Desiccation, that's drying it out. Radiation, filtration. These are some methods that we use to control microbes. And so effectiveness of heat, mode of action, relative effectiveness of heat. You have moist heat. So you have lower temperatures and shorter exposure time than dry heat. So it's relative to dry heat. So you have coagulation and denaturization of proteins, which halts cellular metabolism. So when you have uh, moist heat, it doesn't have to be as high as if you have dry heat. So with dry heat, you have moderate to high temperatures. It causes dehydration, alters protein structure, and also you can have incineration. So moist heat and dry heat can actually be effective antimicrobials. Moist heat is more effective because you can work with that with lower temperatures. Here's some examples here, comparison actually of the times and temperatures to achieve sterilization, moist and dry heat. So remember, sterilization is you kill all microbes, vegetative and bacterial endospores. So with moist heat, if you have it at 121 degrees Celsius, which is about 250 degrees Fahrenheit, it would take about 15 minutes being at that temperature to kill all endospores and all vegetative bacteria. Um, and if it was at 125 degrees Celsius, which is about 257 degrees Fahrenheit, it takes about 10 minutes. So the higher the temperature, the less time it would take. 134 degrees Celsius, which is 273 degrees Fahrenheit, would only take about 3 minutes. That's get really hot. And it would only take about 3 minutes, and it would be able to kill all the microorganisms. That's with moist heat. Now, if you have dry heat, you don't have any water associated with it. At 121 degrees Celsius, just like 121 up here, instead of 15 minutes, it would take 600 minutes. So if it was just dry heat, and 121 degrees Celsius would be about 250 degrees Fahrenheit, you'd have to be there for about 10 hours as opposed to 15 minutes. And then it goes up from there. It gets to the place where 170 degrees Celsius would be about 338 degrees Fahrenheit, and it would take 60 minutes, and that's where you start getting to the place of incineration. Okay. Okay, thermal depth measurements. I need you to know what thermal depth time is and thermal depth point. Bacterial endospores are the most resistant, while vegetative states of bacteria are the least. So thermal death time is usually make a reference to the how long it takes for bacterial endospores to actually die. So thermal death time is the shortest length of time required to kill all the test microbes in a specified temperature. That's the thermal death time. Thermal death point is the lowest temperature required to kill all microbes in a sample in 10 minutes. So if in 10 minutes you want everything killed, what is the temperature? That's the thermal death point. Okay. So methods of moist heat control. So sterilization with steam under pressure. You have an autoclave. So steam must reach the surface of the item being sterilized. The item that you're sterilizing must not be heat or moisture sensitive. So it denatures the proteins, destruction of membranes and DNA. This is one of the autoclaves, like a pressure cooker. Then you can have non-pressurized steam. And so with non-pressurized steam, what happens is that if the substances that can't withstand the autoclaving or the pressure, then what they do is they have steam that is free flowing for 30 to 60 minutes. Then it lets it incubate for 23 to 24 hours, incubate for about a day, and then subject it to steam again. Um, So then you repeat that cycle for 3 days. What we're actually doing with this is since it whatever the substance is cannot withstand pressure steaming, so we can't get it up to a certain temperature, we kill all of the vegetative bacteria in the 30 to 60 minutes. Then we incubate it for 23 to 24 hours. That means that any spores that are there will start to sprout and they will start to become vegetative. So the spores become vegetative. And then after you let them incubate for a day, about a day, then you steam it again. So you kill the vegetative. And you keep doing that for 3 days. So the spores that actually would start to grow that were not killed, then they become vegetative. We kill those. And after 3 days, we pretty effectively have gotten rid of all of the spores and all of the vegetative bacteria without subjecting the item to the high levels that you need to kill spores. So this process is used for some canned foods and also for laboratory media. Then you have boiling water. You have disinfection. Boiling water typically is not able to get to the temperature to kill spores, bacterial spores, and so boiling water is just used to kill vegetative bacteria. So boiling water at 100 degrees Celsius for 30 minutes, uh, and 100 degrees Celsius is about 212 degrees Fahrenheit. For about 30 minutes, it destroys spore-forming pathogens. Then you have pasteurization. Pasteurization, heat is applied to kill potential agents of infection and spoilage without destroying the food flavor or value. They have a flash method, which is 71.6 degrees Celsius for about 15 seconds. That's about 170 degrees Fahrenheit for 15 seconds. That's the flash method of pasteurization. So this is not sterilization. It doesn't kill the non-spore forming pathogens. It lowers the overall microbe count. But with pasteurization, we're not trying to kill all of them. We're just trying to reduce the number. So it does not kill endospores or many of the non-pathogenic microbes. So you have a lot of microbes still in things that are pasteurized. And so that's why milk will go bad even when it's in the refrigerator because it still has microorganisms in it, has spores in it, and they will grow eventually, even in the refrigerator. So milk has a certain shelf life. If you ultra pasteurize milk, which means that you sterilize the milk, you don't just disinfect the milk or reduce the number of microorganisms, but if you sterilize it, you actually kill all microorganisms and all spores. So uh, milk is processed at ultra high temperatures. So that's ultra pasteurized milk, and that's ultra high temperatures, which is 134 degrees Celsius. That's about 273 degrees Fahrenheit. And you do that for 2 to 5 seconds. When it gets that hot, that milk gets that hot, it kills all the microorganisms and the spores. And milk that is ultra pasteurized will last for months as opposed to just a week or so. Okay. So dry heat. So that was moist heat. The dry heat is hot air and incineration. So dry heat uses higher temperature than moist heat. And so with incineration, you have flame, electric heating coil, infrared incinerators. It ignites and reduces microbes and other substances. Very common practice in the microbiology lab. What can you think of in the microbiology lab that we use as incineration? Right, the Bunsen burners, those flames. When we actually put the flame on the loop in the Bunsen burner, then it incinerates any microorganisms that are on it. So it's very common practice in microbiology lab. Then you have hot air or dry ovens, which heated circulated air goes 150 to 180 degrees Celsius for 12 minutes to 4 hours. And that has tendency to coagulate proteins. This is another form of dry heat incineration. This has the infrared incinerator and has a shield to prevent scattering of microbial samples during the flaming. So using heat for sterilization and disinfection, uh, you can use steam under pressure, unpressurized steam. You have pasteurization, and so disinfection of milk and dairy products destroy milk-borne pathogens, and also ultra-high temperatures can sterilize for longer shelf life. And you have fruit juices, beer, wine may be pasteurized, destroy contaminants, and preserve the products. It's important to know that pasteurization, with boiling water, it's limited to disinfection and sanitizing of heat-resistant household objects, such as eating utensils, clothing, sick room supplies, baby supplies. But boiling water is pretty effective to kill vegetative pathogens. Then you have dry heat. You have a dry oven. It's electric or gas chamber. It heats to very extreme temperatures. You do it for 2 to 4 hours. And materials that can withstand hot temperatures and dehydration, like glassware, metals, powders, oils. It's a poor choice for rubber, plastics, and it's also time consuming. Then you have incineration. So the Bunsen burner flame up to 1800 degrees Celsius. That's pretty hot. And so, then you have small infrared incinerators that are up to 800 degrees Celsius. So the furnaces, incinerators can get up to 6,500 degrees Celsius for sterilizing tips of inoculation instruments, limited to high temperature metals. So permanent disposal of waste materials from labs, burning residues, or reduces bulky packages and garbage to ashes. And that's for permanent disposal only, unless it's used on high temperature metals. So the effects of cold and desiccation. Effects of cold is microbistatic, which means it slows the growth of microbes. So it's not microbicidal, which means that it kills microbes. Microbial static means that it slows the growth of microbes. So you can have refrigeration that's 0 to 15 degrees Celsius. 0 is at about 32 degrees Fahrenheit. 15 degrees Celsius is about 59 degrees Fahrenheit. So that's refrigeration. And then freezing, less than 0 degrees Celsius, which is less than 32 degrees Fahrenheit. That's used to preserve food, media, and cultures. So it slows the growth of microbes. But if you notice, if you have food in the refrigerator, it can still go bad. It just takes longer to go bad. So desiccation is gradual removal of water from cells, and it leads to metabolic inhibition. So it's not effective microbial controls because many cells, they retain the ability to grow when water is reintroduced. So uh, lyophilization is freeze drying, and that's preserves things. So which of the following accomplishes microbicidal effects? Microbicidal killing. You can choose more than one. What can accomplish microbicidal effects? Very good. Boiling, pasteurization, and incineration. Freezing is causes microbial stasis, desiccation, microbial stasis. Once you start warming it back up, microbes can reactivate, and once you add water back, microbes can reactivate. So let's talk about some more of the physical methods of control. Let's talk about radiation. Radiation is energy emitted from atomic activities and dispersed at high velocity through matter or space. You have two types of radiation. You have ionizing radiation, that's what the gamma rays, X-rays, cathode rays, and non-ionizing radiation, ultraviolet radiation. This should be in this should be ion this should be an N instead of a D. Non-ionizing radiation is ultraviolet. This is the radiation, just to give you an idea in terms of where we are. This is the wavelength. So this is 100 nanometers to 1,000 nanometers. 10 to the third power is 1,000. In this range is the visible light. This is where we can see. These wavelengths we can see. And they just expanded it out to say between 100 and 1,000. This is where the different colors range. And then above here, which is above 760, you have infrared. That's where here. Above 760, but below 1,000, that's the infrared. Is over there on that side. And below about 380, you have ultraviolet. And that's over on this side, 380 just on this side of 100, yeah, ultraviolet. So then longer wavelengths, you get into the X-rays, gamma rays, those are different wavelengths. And then, shorter wavelengths, you get gamma rays, X-rays. Longer wavelengths, you get infrared and radio waves. So the ionizing radiation. So ionizing radiation has deep penetrating power. It has sufficient energy to cause electrons to leave their orbit. So with ionizing radiation, has deep penetrating power and it ends up breaking DNA. Non-ionizing radiation has little penetrating power. So UV light creates permeating dimers, which means it interferes with replication. So this is ionized radiation. This is a radiation source, and even if there's a barrier, it can actually go through most barriers and and hit the cell, and inside the cell, in the nucleus or in the inside the cell, it gets to the nucleic acids, and it will break parts of the DNA or RNA. So here's shows non-ionizing radiation. With non-ionizing radiation, if there's no barrier, it does the same thing. It causes abnormal bonds in the DNA, RNA. But if there's a barrier, then the UV light does not penetrate, does not affect on the cell. And so here's some of the cellular effects of radiation. This is a normal segment where the adenine, cytosine, adenine, adenine, cytosine pairs with thymine, guanine, thymine, thymine, guanine. What ends up happening is if you have UV light that affects it, then what it does is it creates a thymine dimer. Whenever there's two thymines together, instead of pairing with the adenine like it should, it actually attaches to the thymine next to it. It creates a thymine dimer, and it makes this entire sequence ineffective. So now here's how we use it in common use when we have irradiation. Applications of irradiation control. Here's some raspberries that were exposed to radiation. This 5 days after they were exposed to radiation is how they look. This is raspberries from the same group that was not treated with radiation. And this is 5 days later. See, there's a lot of microorganisms, and they actually had a field day. So applications of irradiation control. In this, it shows some irradiation in water treatment. And it has to be thin layers of water, but it kills the microorganisms on the water without affecting the taste or the quality of the water. So radiation used for sterilization and disinfection. So ionized radiation is sporicidal and penetrating. So there's different medical and commercial uses: cold sterilization, sterilization of foods, including fruits, lettuce, spinach, grains, spices, meats, packaged meats, meals, sterilization of drugs and vaccines. It's rapid and penetrating, prevents infection and spoilage. Then you have non-ionizing or ultraviolet radiation. It is also can be sporicidal and non-penetrating. So with this, you have mostly disinfection of air, some liquids, a few solids. Reduces the airborne pathogens. So as long as it's in an area that, so it's commercial use for clean rooms, food processing areas. So works well on liquids such as water, milk, drugs, vaccines, if exposed to UV in thin layers. Solid materials such as room surfaces, foods, tissues, grafts, drugs, and instruments. It doesn't penetrate through solids, so it limits the application. So it has to have direct exposure. But direct exposure to you, your eyes, and your skin can be detrimental. This is just showing some apples that are being irradiated with cobalt-60 machine that emits gamma rays. And this is a tabletop chamber, generates ultraviolet rays to disinfect a toothbrush. Now you have physical method control, you have filtration. So you have a filter that is so small that it doesn't let viruses or microorganisms through it, but it allows water to go through it. And so these pores are open to allow water to go through. It has a vacuum pump suction, so it sucks it through, and so it actually sucks it down, and now this is sterilized fluid. Filtration, the physical removal of microbes by passing a gas or liquid through a filter. That's what we just described. Used to sterilize heat-sensitive liquids and air in hospital isolation units and industrial clean rooms. So then we have chemical agents in microbial control. We have disinfectants, antiseptics, sterilants, degermers, and preservatives. These are some desirable qualities of germicides. First off, you want it to have rapid action and low concentration. You want it soluble in water or alcohol, and you want it stable when it's soluble in water and alcohol. You want its broad spectrum, which means it can affect a whole lot of microorganisms. You want low toxicity. You don't want it to hurt people or living tissue other than microorganisms. You want it penetrating. You want to be able to get through the things that you need to get through to get to the microorganism. You want it to be non-corrosive and non-staining, and you want it affordable and readily available. These are some desirable qualities. Then you have germicides at different levels. So first off, you have high level germicides. They kill endospores. So they may be sterilants and will sterilize things. And then intermediate level, they kill fungal spores, but not bacterial endospores. So they kill pretty well. They kill the tubercle bacillus and viruses. So intermediate level. So they can kill quite a few things in vegetative bacteria and all, but they will not kill endospores. And so these are used to disinfect devices that will come in contact with mucous membranes, but are not invasive. So then you have low level chemical decontamination, and that's low level will eliminate only vegetative bacteria and vegetative fungal cells and some viruses. So use these on clean surfaces that touch the skin, but they don't touch mucous membranes. Here's some factors that affect germicidal activity of chemicals. First off, the nature of the material being treated. So the degree of contamination, how contaminated it is, time of exposure to the microbial agent is required, and then concentration of the chemical agent. So the strength and chemical action of the germicide expressed in various ways, like the dilution factor or dilution percentage. So these are some of the factors that affect how effective a germicidal chemical will be. Here's some of the qualities of chemical agents used in healthcare. We have chlorine, iodine, phenols, verhexidine. I'm not going to have you know these in particular, but it's good for you to kind of understand some of these. Like with chlorine, it's highly toxic and the solution irritates skin. So its level of germicidal activity is intermediate and it's inactivated by organics, which means that bodily fluids and tissues will kind of make it inactive. It's unstable in sunlight. Okay. Then you have alcohols. Target microbes and most bacteria, viruses, fungi. It's intermediate level, and it's flammable, fast-acting. It's toxic if ingested. A mild irritant, so it dries your skin up. Then you have hydrogen peroxide. It's a sporicidal, which means that the level of germicidal activity is very high. But it's toxic to your eyes, toxic if ingested. It works well in organic matter, though. Then you have the quaternary ammonia compounds, low to intermediate. Weak solutions can support microbial growth. So if you have a weak solution of it, microbial growth can actually occur. It's easily inactivated. Then you have soaps. It has very low germicidal activity, but it's non-toxic, few if any toxic effects. It's used for removing soil, oil, and debris. So that's what we talked about in terms of degermination and also sanitation. So you can reduce the load of microbials by using soap. So mercurials, easily inactivated. You don't see these too much. It's very low as well, or low. Okay, I'm not going to ask you the different chemical agents, but glutaldehyde is a sporicidal. It's very high sterile. Irritate the skin, toxic if absorbed. It's not inactivated by organic matter, but it's very unstable. And then you have this is a newer one. It's less toxic than the glutaldehyde, highly stable, but it stains the skin. So we have some things that we have that can actually kill the spores. Um, when you see a star, if you go back to those tables and you see the star, these chemicals approach the ideal by having many of the following characteristics. Broad spectrum, which means it affects a lot of microorganisms. It's not toxic, not very low toxicity. It acts very fast. You don't have to let it soak for a long period of time. It has good penetrating ability, has residual effects. If it's on the surface, it'll keep killing. It's very stable. It's potent even in organic matter, and it can be diluted. Okay? So here's some of the germicidal categories. So let's talk about the halogens, like chlorine bleach, denatures proteins by disrupting disulfide bonds. So it's intermediate, unstable in sunlight. So we use it in water, sewage, wastewater, inanimate objects. So you use it on non-living surfaces and substances. Chlorine. You have iodine. Interferes with disulfide bonds of proteins. Intermediate level does milder medical and dental degerming agents. So you use this in surgical scrubs.